So this whole presentation is being delivered by Team 2, and our topic for lesson 11 is on insulin signaling. So this slide is talking about the definition of insulin signaling. Insulin signaling starts with the hormone insulin binding to its cell surface insulin receptor, which is protease kinase. Next, one or more phosphate groups are added to the insulin receptor kinase, and the receptor kinase is activated to tyrosine phosphorylase, important cellular substrates that are critical for entraining the insulin response. Insulin signaling is a process that is started through binding and activation of the cell surface receptors and starts a series of phosphorylation and dephosphorylation events, second messenger generation, and protein-protein interactions that lead to different metabolic events in nearly all tissues of the body.
Insulin is a hormone that is secreted by beta cells of pancreas, which is composed of two polypeptide chains linked by disulfide bond. Insulin, which is a signaling molecule, binds to a receptor tyrosine kinase, then it will travel through the bloodstream. Insulin signaling is considered long distance, which is endocrine, as it releases insulin throughout the body, meaning more target cells that are further away from each other, like in muscle and fat cells, instead of neighboring cells like paracrine signaling.
The phosphatidylinositol-3 kinase pathway, or the AKT pathway, is a signaling pathway that plays a part in regulating various cellular processes like proliferation and cell growth. It can be activated through various mechanisms, including hormones, growth factors, or the extracellular matrix. A ligand will bind to cell surface receptors, such as receptor tyrosine kinases or G protein-coupled receptors. After activation, PL3K phosphorylates the phosphatidylinositol 4,5-biphosphate to generate phosphatidylinositol triphosphate. It is a second messenger that recruits AKT, which is protein kinase B, to the cell membrane. AKT is then activated through phosphorylation by PDK1 and the mammalian target of rapamycin complex 2. Activated AKT then phosphorylates a variety of downstream targets, including proteins involved in cell cycle regulation, like for example BAD. In the pathway, there are some loops that help to regulate its activity and prevent excessive signaling. Activated AKT phosphorylate and inhibit the activity of upstream components, such as RTK, which is receptor tyrosine kinases, to weaken the signaling cascade.
Okay, the next thing we're going to talk about is the RAS/RAF/MEK/MAPK, which stands for mitogen activated protein kinase pathway. So this pathway is a central pathway which regulates cell growth, proliferation, survival, and differentiation. This signaling cascade is essential for intercellular and intracellular communication, and is activated through extracellular signals, which are the growth factors, or the hormones, or when a signal binds to the protein tyrosine kinase receptor. The RAS activates the pathway and transmits the signals downstream, resulting in the transcription of genes involved in controlling several cellular mechanisms. The ligand binding to the receptor leads to the activation of the cytoplasmic tyrosine kinases, short form for TKs, which phosphorylates tyrosine residues at the cytoplasmic tails of the receptor. So this activation converts GTP to GDP on RAS and leads to the phosphorylation of the downstream signaling molecules. And the RF protein kinases phosphorylase and activates the MAPK kinase, and the MAPK translocate to the nucleus, activating transcription factors leading to the changes in gene expression.
Lastly, for the components in signaling pathway for insulin, under the reception stage, you have your insulin, which is the substrate, and it is secreted by beta cells in the pancreas. After that, you'll bind to this receptor called tyrosine kinase, which is an example of your enzyme link receptor, as it phosphorylates itself at each tyrosine residues. After that, you'll move on to a transduction stage whereby you have your IRS, which is a transmembrane glycoprotein, and it recruits and binds to PIK3 to signal transduction. And your PIK3 is actually an enzyme kinase that catalyzes PIP2 to PIP3 and acts as a second messenger which increases concentration to relay and amplify the signal to activate AKT. And your AKT is the last enzyme involved, which is to inhibit GSK3 activity and increase glycogen synthase or glycogenesis. And the other function is to activate other substrate, for example, like GLUT4, which is found in your response stage, whereby it is a protein translocated to uptake glucose into the cell for glycogen synthase, which happens in a plasma membrane. So all these components come together to fulfill the function of an insulin, which is to bring down the blood glucose level.